Now we're going to look at phase 2 of glycolysis, the energy recovery part of this metabolic process. Again, here's that general overview. We start with 2 glyceraldehyde 3-phosphate molecules. Remember we get 2 of those from reaction 5 in phase 1. In this conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphate, we have an inorganic phosphate added to the gap molecule. We also have the involvement of NADP, which picks up 2 electrons and generates this high energy compound, NADH. Glyceraldehyde 1,3-bisphosphate glyceraldehyde, then is capable of phosphorylating an ADP molecule to generate ATP. So for each gap that goes into phase 2, we're going to produce 2 ATPs, or a net of 4. When we subtract the 2 from phase 1, we have an overall gain of 2 ATP. This process, the phosphorylation of ADP, is called substrate level phosphorylation. In the next step, we have a rearrangement. So we're going to move that phosphate from the 3rd carbon to the 2nd carbon. So now we have 2-phosphoglycerate. Again, that was a rearrangement. This next conversion, we're going to generate a high energy compound called PEP, phosphoenolpyruvate. And this molecule will then donate its phosphate to another molecule of ADP to produce ATP. And we get the final product of glycolysis, our pyruvate molecule. I want you to stop this now. Take time to look at the video where we draw these, and draw these with me please, molecules together, discuss the enzymes, cofactors, just like we did for phase 1. Then please come back and finish this video, which goes on to discuss other fates of pyruvate and other carbohydrates that enter into glycolysis. So now we're going to look at alcohol fermentation in yeast. Our next topic will be the citric acid cycle, into which under aerobic conditions the pyruvate is processed or shunted to produce more energy. We're also going to look at anaerobic conditions with respect to lactate. But right now, let's take a look at alcohol fermentation. So during strenuous exercise, muscle tissue becomes anaerobic, so it uses up the oxygen. And the oxidation of acetyl-CoA is limited then by the availability of oxygen. In this case, pyruvate is diverted into a side reaction that utilizes lactate dehydrogenase to produce lactate. So this is a reversible reaction. We have pyruvate in our muscle tissue. It's converted then to the lactate. NADH is then going to be oxidized to NAD+. And we'll talk more about these 2 forms of this molecule as we move along. Excuse me. The lactic acid, by the way, can be directly converted back to pyruvate, which then can go into the citric acid cycle. I forgot to tell you that. So, alcohol fermentation occurs in yeast, as we've seen before. We, animals and plants, do not have the enzyme pyruvate decarboxylase. Thus, we cannot ferment pyruvate. So the pyruvate undergoes a decarboxylation, so that removes a molecule of CO2 and forms acetaldehyde. Here we have NADH, that high energy molecule that carries 2 electrons. So this process is going to be a reduction of acetaldehyde to ethanol. NADH is going to lose 2 electrons. It converts back to NAD+. This occurs by the action of alcohol dehydrogenase, and the end product then is ethanol. Animals do have alcohol dehydrogenase, and this is a liver enzyme. And we will take a look at a case study that looks at the reaction of alcohol dehydrogenase. So this figure shows the glycolytic pathway, and the primary substrate for that is glucose, as we have just seen. However, our typical diet contains carbohydrates other than glucose and its polymers. So for example, milk products contain lactose, which is a combination of glucose and galactose. So galactose can be phosphorylated on C1. So it undergoes a phosphorylation and then an isomerization to glucose, and thus glucose 1-phosphate is isomerized into glucose 6-phosphate, and then enters the pathway the same way glucose would. Sucrose is a combination of glucose and fructose. So that glucose would just enter the pathway. Fructose, however, undergoes a phosphorylation followed by a cleavage. So we get diglyceraldehyde and dihydroxyacetone phosphate. So that's from that C1. That glyceraldehyde phosphate gets phosphorylated by triose kinase and enters as glyceraldehyde 3-phosphate. Mannose is phosphorylated by a hexokinase on C6. It isomerizes and enters as fructose 6-phosphate. We're going to talk more about the entry point for fructose and the concerns about the increased fructose consumption in our diet, which may contribute to greater production of lipids and fats and, unfortunately, obesity.